All right, are we ready to talk about viruses? I am not going to do viruses justice. We're zooming through this. We're like, literally, what are the top 2 highlights that you need to know? That's about it for this. If you like viruses, they are very cool. Take Med Micro, take biology of viruses. I'm sorry if I don't prepare you well enough for biology of viruses, those of you going into that. If we have more time near the end, I'll bring back some of the virus stuff. When we get to the disease stuff, they'll probably fit in a lot better there. Okay, viruses. Let's get to it. Not too many key terms for this one. What is a virus? It's a non-cellular particle. It's not a cell. Cells have cytoplasm, they have DNA or RNA, they have ribosomes, they have a plasma membrane. These don't have them. They got DNA or RNA, and they have protein to cover it, and that's it. They can't do most of the things that life does. If you look at like the characteristics of living things, they only do about like 3 of them. You need to do all of them to be considered alive. So they infect a host cell and steal our machinery. They can't do any metabolism on their own. They can't make proteins on their own. They need our stuff to do it. So they come in, they sneak in, and they use our stuff. Their whole goal is to make more virus. I mean, that's the goal of all things, really, make more of ourselves. But viruses do it like crazy. They are inert. That means they have no metabolic capabilities on their own. Now, the particle itself is called the virion. If you ever hear that term, that's what it means. I tend to just use virus because it's just a little easier, so I might go back and forth there. It's usually DNA or RNA for the genome. It can be either. We'll talk about the types. And it's surrounded by protein. No cell membrane, unless you're a very, very special kind, and it's not your membrane. You stole the membrane from somebody else. So really, protein, DNA or RNA. Right. So viruses are very specific to who and what type of cell they can infect. There are some viruses that infect just one species, not even just one species, only one type of cell in that species, like rabies. It infects our neurons. It doesn't really give a crap about your epithelial cells. It cares about your neurons. So ones that infect bacteria, are the ones that we're going to care about a little bit more, are called bacteriophages or phages. And we're going to see them in the lab. It's actually kind of cool. We'll mix them in with bacteria and have the bacteria grow, and you'll start seeing these little circles where all the bacteria are dying out and it becomes clear, and that's where the phage is multiplying and killing the bacteria. And we call those little empty spots plaques. You can kind of see them on that picture there. And human viruses, we know a lot of them. Plant viruses, we're not going to talk about too much, but we talked about the tobacco mosaic virus. There's pox viruses for plants. They got lots of crazy viruses too. All right, viroids are interesting. They're not quite viruses. What they basically are is just RNA. They have no protein capsule. So these are infectious RNA particles. They get into the host, and the host's RNA polymerase will copy them. And they're spread from cytoplasm to cytoplasm. So if 2 cells are kind of floating around, they're fine. But as soon as they touch, that viroid can jump in. And they really messed up the translation process for the host. RNA is cool. We'll probably talk about it a lot more in our DNA section, but RNA is way more versatile than DNA. It can act like an enzyme, and it can act like a nucleic acid, so it's really cool. And they will act like those enzymes and do crazy things in the cell. Now, the opposite of an infectious just RNA molecule is an infectious protein molecule. So if we don't have the genetic component and we just have the protein component, this can actually cause infection too. These are misfolded proteins, very specific ones. Not if a random protein just misfolds a little bit, not necessarily a prion. These are very specific proteins that misfold in a very specific way, and then they can catalyze the misfolding of other proteins. So if this protein bumps into another protein, it misfolds and it's now a prion too. These are scary. We don't really know how to treat them. They're usually kind of death sentences, so don't eat brains. That's what we often do. They like to live in our nervous tissue. So one example is bovine spongiform encephalopathy. You guys might be a little young for this, but mad cow disease when I was a kid came into the world like crazy, and it was really scary for a while because everyone's like, I'm going to eat a cow and I'm going to get mad cow disease and die. It's still out there. There's scrapie in sheep. There's Creutzfeldt-Jakob disease in humans. A lot of times you get these from eating infected prions, and then they get into your system, they affect your brain, and you die. Something like Creutzfeldt-Jakob disease, though, it can happen spontaneously. You can just all of a sudden one day have the protein misfold in just the right way, and now you have the disease. So they're kind of scary. There's not a lot of classes that teach about prions because we don't know much about them. So let's talk about the structure of a virus. The whole point is to protect the genome long enough to get it into the cell. This is actually the smallpox virus right there, which is really cool. They have a very, very, very small genome. So these guys don't carry a lot of information. They carry just enough to make more viruses and to make the structures that they need to get in the cell or get out of the cell. And their protein capsid is usually made of 1 to 3 different proteins. Once again, they don't have a lot of space to make more proteins than that. So they'll just make a lot of copies of the same structural protein and use it to make the capsid. The capsids can have really cool shapes. I'll show you one of them in a second. And sometimes they're symmetrical, sometimes they're asymmetrical. We're not really going to talk about that too much. The book goes into like filamentous viruses and icosahedral viruses. We're not going to get into all that here. It's cool stuff if you care about it, though. Give you like some fun reading after the class ends. Some viruses can have an envelope. This is a lipid bilayer, a membrane. It will surround the capsid. They don't make it. They will push out of the cell like they're kind of budding out of the cell and steal the membrane with them. Sometimes it's the plasma membrane of the cell, sometimes it's the ER membrane of the cell. It just depends on the virus. What this does is it basically tells the immune system, I'm not a virus, I'm you, so don't attack me. And it helps them get into our cells easier as well. That's a very, very surface level explanation of an envelope, but yeah. The problem is these guys are more sensitive to environmental stress because membranes are more sensitive to things than proteins are. So their genome is cool. It can be DNA or it can be RNA. Having a genome made of RNA is weird. And it requires a lot of different adjustments to the central dogma of biology, which we'll talk about. It can be single-stranded or double-stranded. You can have single-stranded RNA or double-stranded RNA. You can have single-stranded DNA. All types of viruses. They can be linear, they can be circular, or they can even be segmented, just like little pieces of DNA. They're just kind of floating around inside the capsid. And we use their genome to classify them, which I'll talk about a little bit later. And small viruses have small genomes, which makes sense. So sometimes the way they make their genome even smaller is they overlap their genes. It can basically be one sequence, depending on where you start the reading frame from nucleotide 1, 2, or 3. You can have 3 different proteins in one sequence. And that's what they like to do. They like to overlap it a bit so they can condense space even more. Now, bacteriophages, these guys are cool looking. They look like little spiders. This is literally their protein capsid, this whole thing, and inside here is their DNA. Their genome, excuse me. All of this serves to get the genome into the cell. This big guy here is a bacteria, and all these little guys on the outside are the bacteriophages trying to get their DNA or their genome into the cell. They have tons of diversity, so not all of them look like this, but a lot of them do. And we're going to talk about how they replicate inside the cell, because there's 2 main methods. So to start an infection, the phage has to first contact the cell, obviously. It can't infect you if it's far away from you. It has to physically touch the cell and physically inject its genome inside. Oftentimes they'll have really cool structures that basically act like a needle and poke through the entire cell wall. So that's a thick structure to get through. And the way that they attach is by finding all these different surface things on the envelope, like a flagella, like an efflux pump, like a porin, like LPS, and it will attach to those and find a way to stay attached. So if these start to change their structure, the virus won't be able to attach anymore. So the virus will then have to evolve to be able to attach to this new form of OMPF or whatever it is. So it's basically like always an arms race. The bacteria evolve, then the virus evolves in response, then the bacteria have to evolve again. Evolution of diseases is so fascinating. If you're curious about that, once again, med microbiology of viruses, fun stuff.
Really good question. So if it's not alive or we don't describe it as alive, how does it know where it's going and that?
Yes. With all biology, I keep anthropomorphizing it. It wants to do this. It's going this way. This is all random molecular motion. So things just vibrate and move, and they happen to hit each other. This guy could hit this bacteria and not attach. It has to hit in the right way with the right speed and everything for the chemical reactions to occur. This is all just random chemical interactions. It blows the mind if you think too hard. Don't think too hard about it. That's like a 2 a.m. I'm drunk kind of thinking process. Not now in class. The sun's out. We can be happy. All right. So they use like a needle to inject their genome. Most bacteriophages are DNA genomes. So they don't have a lot of crazy mechanisms that they have to employ. So now the capsid has injected the genome, and it's just a protein shell. So we call that a ghost. And if you guys remember from 106, 107, the way we learned that DNA was the genetic material was because Hershey and Chase, they labeled either the DNA as radioactive or the capsid as radioactive, and they looked where the radioactivity moved to. If it stayed on the outside, that with the protein's obviously not infecting the cell. But the DNA, if the radioactivity moved in with it, that means DNA was the infecting thing. That's the genetic material. So once again, microbiology taught us about biology in general. We learn everything because of microbes and viruses. So let's talk about the two cycles once they get into the cell. Now, these aren't mutually exclusive. Sometimes cells can only do the lytic. Other types of cells do the lysogenic cycle and then transfer into the lytic cycle. Just kind of depends. So a type of virus that can do the lysogenic cycle is called a lysogenic phage or a temperate phage. If you hear those terms, that's just what they mean. The decision of which one they choose, if they can do both, is based on the environment. Once again, chemical cues telling it what to do. And basically, the main thing that will trigger a cell to go from the lysogenic cycle into a lytic cycle is stress. The host cell is being threatened. I need to get out of here before I die too. So let's talk about these two different phases. It'll make more sense then. So the lytic cycle is the most direct and the most immediately destructive. The virus enters the cell, it puts in its DNA. The DNA has to circularize itself. Bacteria like to keep their genome and all their plasmids in circles. So they've learned over the years, if something linear comes in, attack it because it's not us. So the virus learned then over the years, okay, I inject my DNA and it has to circularize or it's going to get attacked. So once it circularizes, they start to use the cell's machinery, the cell's RNA polymerase, the cell's ribosomes to start making more phage. You copy the genome, you copy your proteins, you assemble them, and then they exit. And the way they exit is by basically bursting out of the cell. Most of the time they will lyse the cell and the cell will die. That's why we call it the lytic cycle, because it's lysing the cell, breaking the cell open. The idea is get in and start replicating as fast as possible, because we're going to learn later, bacteria do kind of have sort of an immune system that they use to destroy and target viruses. So they want to evade that, and they want to replicate as soon as possible to then go infect more things and replicate more. So they get in, they replicate, they burst out of the cell. I am a nerd, I'm sorry. All my references are sci-fi fantasy. Sorry about that. So integrated viral genomes. Now, to understand the lysogenic cycle, we have to know that viruses can take their genome and stick it into the genome of the cell that they're infecting. They can say, I'm going to sneak it in here, and it's going to be a part of you, and you're not going to want to destroy it now. So in this case, a phage that integrates is called a prophage. In humans, we call it a provirus. Something that permanently integrates is called an endogenous virus. 8% of our non-coding DNA is viruses. 8%. Now, some of them are no longer active. They've mutated and they don't work anymore. Some of them potentially do, and we don't know what they do. Maybe they have an immune system function that defends us from bad bacteria or from other viruses, who knows. But they integrated and they stayed. The way they stay, if I have a virus come in and integrate into a skin cell of mine, oh well, when I die, that virus is gone too. But sometimes they'll get in and they'll integrate into the DNA of the egg or the sperm, and those are then permanently integrated in any child that you have with that egg or that sperm. Then they will just have it in every single cell of their body, and that means when they make egg and sperm, it'll pass on to them as well. It's crazy stuff.
Isn't that like HIV?
No, yes, HIV infects our white blood cells, though, our T cells. So it won't really go into our germline cells, our egg and our sperm. It's a little bit different. So yeah, it does infect cells and integrates in, but not in that way. So you can't pass it on to your kids. Not that I know of. It seems like all the ones that we've learned at least a little about are so ancient that we can't figure out what their function is anymore, or we think they have a protective nature. Otherwise, they'd just hurt us, and we would try to expel them, or that person wouldn't live long enough to pass them on.
Do you think some
Maybe. We do know that sometimes when they come and they integrate, they bring in new genes that help us do something. So a lot of evolutionary leaps that organisms have taken are because viruses bring in cool new genes for us to play with too. So if they can bring in good ones, they can also bring in bad ones. This is also how we get viruses that cause cancer. They insert into genes that control mitosis and make our cells divide out of control, or they insert in such a way that it breaks up a key protein and now our cells start to get sick and maybe we get an autoimmune disorder. It's a possibility. I don't know of any examples where a virus has triggered that. I'm sure they exist, and I just don't know them. All right. So the lysogenic phase has to deal with this integration into the genome. So they enter into the cell in the same way, and then once the virus genome is inside, it will integrate, and then it will stay in the cell. It replicates by the cell replicating. So it wants that cell to live a long and happy life and make lots and lots of baby cells. Because the more baby cells it makes, the more virus is being protected and being made. But if your cell suddenly starts going through a starvation phase, or it's being attacked by something else and its survival is being threatened, it can trigger entry into the lytic cycle. And it will all of a sudden enter the lytic cycle, immediately start replicating like crazy, make new viruses, and burst out. So it can do both, and it decides, based on the environmental cues, which path it takes. Some Some that can do this lysogenic cycle will just come in and immediately do the lytic cycle. They don't have to go into the lysogenic cycle. It's all decisions, chemical decisions. Any questions about that?
So it can connect and then disconnect.
Yes. Mm-hmm. It can integrate and then be pulled out of the genome. Or it can integrate and stay and still make new phages while it's integrated. It just depends. Sometimes they'll cut themselves out and bud out of the cell, so it won't kill the cell. It will just remove itself entirely and make a new little virus. That's more in like human and plant viruses. But yeah, that's, with bacteria, it's usually stay inside or kill the cell. All right. I'm not going to talk about this right now. So this slide, cross it out. I won't ask you about that one. We'll talk about this stuff more too when we get to the DNA stuff. We're going to get really deep into DNA. So as I mentioned, often this going back into the lytic cycle can be random or it could be environmental stressors. Bacteria have defenses, lots of them, against phages because there are lots of phages everywhere. What do they say, like 1 drop of seawater contains like 1 billion bacteriophages or something like that? It's an insane amount. Maybe it's like 1 milliliter of seawater. But bacteriophages are literally everywhere all the time. So we can mutate as bacteria to alter our receptor proteins. If you can't bind to me, you can't inject your genome. This is random. The mutation has to randomly occur. They can't be like, I'm going to change and now I'm changed. We can't mutate our DNA on like that, which would be cool. So we also have these restriction endonucleases. So these will cleave DNA and RNA. They look for DNA that doesn't look like theirs. There's a lot of ways that they can tell. One is it's linear instead of circular. Sometimes they look for very specific patterns that they've recognized as that's only in viruses, not in us. And then they'll cut it all up. Another way they do it is through methylation. This is something we're learning more and more with epigenetics. We can methylate or acetylate our DNA, and it causes the DNA to either coil up tighter or loosen up. And that determines how we can replicate our DNA, what genes we can actually access to turn into proteins. So we modify our genes as a result. And bacteria, and we do this, viruses can't do this. So if they see DNA that has no methyl groups on it, they're like, that's not us, and they'll attack it. The other one is CRISPR. This is chapter 12 that really gets into it. I want to talk about it with you, and I'm going to try my best to because it's really cool stuff, and a lot of people are fascinated by it. But we might have to like talk a little bit about it here, more there, and do it in little pieces to help save time. But basically, this is the bacteria's immune system. It's basically like our T cells and B cells that remember and can go out and directly target specific viruses. So they recognize very specific patterns of DNA, and they go in and they cut it. And if it's cut and it's broken into little pieces, we can degrade it with other enzymes, and now it can't affect us anymore. And we've taken advantage of this. Since it recognizes a specific sequence, we can give it a sequence to recognize, and we can then put it into our cells and say, hey, this person who has this bad cystic fibrosis gene, cut out the bad gene and insert the good gene for us. And now the person won't have cystic fibrosis anymore. This is very, very, very new to use it in humans, but bacteria, it's been around for millennia, potentially millions of years. It's an old thing for them. Animal viruses, way more complex. They have way more complex attachments. They have significantly more specific tropism, which is, I think, here, what specific things they can bind to and infect. Some of them have to do uncoating and release that membrane that comes with them. So animal viruses are just a lot more specific. That was the point of those two slides. We do classify viruses based on their genome composition. Do they have RNA or DNA? And how do they express their mRNA? I'm going to touch on this very, very briefly. I will not test you on any of this. But it's cool, and I think you should understand it because it is important and it primes us for our DNA stuff. So there's 7 groups of viruses. The first one, group 1, double-stranded DNA viruses. These are normal. This is basically like, yeah, you put in DNA and the cell knows what to do with double-stranded DNA. But the rest of them, we're kind of breaking the central dogma to some extent. The central dogma is DNA replicates itself, DNA is transcribed into RNA, RNA is then translated into proteins. If we want to take RNA and replicate RNA, that's a violation of the dogma. If we have single-stranded DNA, kind of an alteration of the dogma. If we want to go from RNA back into DNA, that's definitely a violation of the dogma. So these viruses break this rule all the time. And the reason why this exists is because it's based on the enzymes available. Most living things only have enzymes that can work on double-stranded DNA. They can only make RNA based on a DNA template. They can't make RNA based on an RNA template. So in order to break these rules, we have to create new enzymes to break the rules, and viruses can do that. So DNA viruses basically use all of the host stuff because it's the same as what we use, DNA, double-stranded DNA. RNA viruses, the big thing with them is they need an RNA-dependent RNA polymerase. Our RNA polymerase is technically called DNA-dependent RNA polymerase. It needs DNA to make the RNA. This one can make RNA using RNA. Then we also have retroviruses, which need reverse transcriptase to take RNA and turn it into DNA. And sometimes then it'll incorporate itself into the genome after it's turned into DNA. So if you're curious, I went through all the different groups and explained how they do this, gave you some examples. I will not test you on all this, but you can read through it because it is quite fascinating, and it does really help you prime your brain, as I said, for the genetics talks that are coming up. Okay. And then you got some study questions. Mainly focus on the lytic and the lysogenic cycles when you're studying and how, the structure of the virus and things like that. Okay. Let me stop the recording. Take some water. And then I'll talk about